I'll introduce our speakers, and this will give folks a chance to do the continuing education link as well as um, as well as what's going on? Do the geography. John is from the Midwest, grew up in Michigan, earned degrees in natural resources and entomology and insect pathology, finishing up with a PhD at Oregon State University. He worked as a research entomologist with the USDA Agricultural Research Service since the early 80s, and for the last 17 years has worked in Ithaca, New York, which is how I came to know John. He's worked on pathogens of both pest and beneficial insects since 1975. And in Ithaca, his work has been focused on developing safe, effective alternatives to chemical pesticides for pest management. Currently, the target pests he studied are those of emerald ash borer and 2 species of Asian ambrosia beetles. And he's an adjunct professor in the Department of Entomology here at Cornell and is active in professional societies and on scientific journal editorial boards. Leah is from Massachusetts and is now currently based in Michigan. She received her bachelor's degree in natural resources from the University of Michigan, entomology, master's in entomology from University of Maine, and PhD in entomology and parasitology from the University of Kentucky. So both John and Leah bring us insect and pathogen expertise. She's worked at the Leah has worked at the USDA Forest Service Northern Research Station as a research entomologist since 1979 in Minnesota and now Michigan. She's an adjunct associate professor of entomology at MSU and has maintained a laboratory there for over 20 years, doing research involving methods for using microorganisms and insect parasitoids for the management of invasive forest pests. Since 2002, she's worked on emerald ash borers, natural enemies, both in Michigan and in China. And her research in China and in her quarantine lab has led to the development of an interagency EAB biological control program that started last year. She continues to work closely with university and federal researchers on methods to conserve ash species, the genus, in forest ecosystems. So we have 2 outstanding scientists here who've been doing some great work and have been willing to share that. So it's to our benefit. And I see that we have people have continued to have signed in from Vermont and Nebraska and Maine and Pennsylvania. So there's a large number of people from all around the Northeast. We're going to jump over to the presentation screen, and John and Leah are going to start with. John, let's see.
I can load it up. I didn't know if you were gonna. I'll
Oh,
start
there
with
we go.
you
The
got it.
intro. Okay.
Right.
So let's bring that in, do this. And let me scroll it over.
Perfect.
Hang on. We're not quite there.
I think there's a fit page button at the top, Peter.
Okay.
The thing with 4 arrows. There you go.
There we are. That's what I was looking for. Okay. And we're still dangling off the edge here. Let me move us over a little bit. Okay, with that, I'll introduce, or I'll actually turn the floor, as it will, over to John and Leah. Welcome them, and I'm going to turn my microphone off and let you guys have at it with the audience. So welcome. Thanks for your attention to working on this webinar, and I look forward to your presentation.
Thank you, Peter. And on behalf of Leah, I thank all of you for being here. It's an honor to be invited to participate and to be able to have this relatively new outreach tool at our disposal. I first met Leah through my sister, whom she befriended when we three were all undergraduates at the University of Michigan in the 1970s, and Leah and I overlapped as master's students at the University of Maine. So she's a longtime friend and colleague, and it's a pleasure to have this chance. I think this is the closest our research careers have intersected, and that is our various missions to work on the emerald ash borer. So I'm going to start, then Leah's going to take the bulk of the work talking about parasitoids. And if time allows, I'll tell you about a piece of research that the two of us together with some collaborators are working on in Michigan, testing a microbial insecticide. First, I thought, since I know many of you are from New York, I'll tell you my own story of having been one of two, along with Mike Griggs, to discover emerald ash borer in New York about a year ago. And that will give us a chance to review the biology of the critter and talk about the signs or symptoms of its infestation of ash trees. And so we all know, I think, many of us, that this is Agrylus planopenis. It's a gorgeous beetle. Let's see if I can figure out this pointer, which I think I have at my disposal somewhere. No, maybe that's a PowerPoint thing. Oh, there it is. I got it. Thanks, I see it. All right. Now, there's a green arrow on the screen, but I don't know if I can move it around. Here we go. Yes, I can. So when the beetles, I'm giving up on that, y'all. I'm getting used to this. The beetle on the lower right, when they flex their wings, the top half of their abdomen is kind of a gorgeous burgundy color. The beetles there on the penny are dead, and they've lost some of their luster. A reminder about its life cycle. Adults chew their way out of the tree in early summer or late spring, as you can see on the upper left, and they leave behind a characteristic D-shaped hole. They don't leave behind the white spray paint that marks this one, though. It takes females 2 to 3 weeks of feeding, during which time they mate, before they are indeed gravid or able to lay viable eggs. And so there is a window of time after emergence for any individual beetle when it's not doing us much harm except chewing on leaves. And that might be a time to target things. The eggs are tiny. You can see the scale bar there, a millimeter in diameter at most. They tunnel right into the tree and do their thing. On a newly infested tree, on average, the beetles might spend 2 summers as a larva, and be in a 2-year life cycle. In more heavily infested trees or higher density populations, the life cycle can take place in 1 year. The larvae overwinter. They develop as larvae. They chew their way into the wood and sort of make a U-turn and spend the winter as a fully developed larva. They pupate in the spring, as you can see in the lower left, and they position themselves as chewing larvae in such a way that, as newly developed adults, they don't have a whole lot of the tree left to chew out of. They do most of their chewing as larvae, and the adults just need to get out when they get there. Another review, I suspect for many of you, the beetle is native to Asia, was found near Detroit in 2002, probably arrived in wood packing materials. There is a large volume of wood called dunnage, D-U-N-N-A-G-E, traveling our oceans as ballasts or as like packing peanuts in the holds of cargo ships. And that, thanks to the work of one of Leah's colleagues at East Lansing, Michigan, Bob Hack, and others, there is an attempt to regulate that as it's not really a commodity, but it's important, and it's a likely source of invasive beetles from one continent to another around the globe. And that's a possible way that emerald ash borer got to the United States, perhaps the port of Detroit or Windsor, Ontario, or Toledo, somewhere there. Only ash are affected. Many millions and billions of trees are dead by now, and the beetle has now spread to 14 states and 2 provinces. I'll show you a map here in a second. Yes, so this map is, you can see up on the upper right, the bottom there, the date of April 1st, no fooling, 2010. It's incomplete because the beetles have now been confirmed in Iowa, and more counties in Pennsylvania have been officially confirmed. And so, and you note in the upper right, parts of Quebec, I believe that's near Montreal, and the part nearby in Ottawa, or near Ontario, thank you, is near, somebody's on the arrow, but not me. That's near Ottawa, Canada's capital. So all of those states are now infested, and the blue lines around those areas signify federal quarantine areas. Other colored areas mean different things, and the map is worth studying if you're interested at www.emeraldashborer, all one word, .info. Okay, so it was a year ago. It was a Sunday. So Mike Griggs and I have been going back and forth to Michigan an awful lot the last handful of years. And on one of those days coming home on Interstate 86 near Randolph, we were zipping along at 75 miles an hour. And Mike, I was driving, and Mike said, "Ooh, look at those trees, and we got to stop on our next trip out." So that was a Thursday. Sorry, I said Sunday. That was Thursday, the 11th. And on Sunday, the 14th, on our way back to Michigan for the next week's worth of work, we did stop. And we were on the westbound exit ramp of Interstate 86. We parked off the shoulder. We walked down. We looked to the right. We saw this tree. We look to the left. We see this tree. We look straight ahead, and we see this. It's at a truck stop there. You won't find these trees anymore, but and so this tree, I have a couple more pictures, as I took these after we'd already scraped into the bark, but and if you look at the branch coming horizontally toward you on the right-hand side, there's a picture of that next. Without us having to damage the tree in any way, we could walk up at eye level and look at that, and you can see the characteristic galleries left by emerald ash borer. So at that moment, we knew. It didn't take long for us to see. This is a Where's Waldo picture of where are the exit holes, and I can see at least 2 in this photograph. The D-shaped holes that I'm sure you can pick out. We saw those in the bark, and you know, at one moment, Mike Griggs said to me, "Hold still," and he tried to grab a critter off my shirt, and sure enough, we were looking at the emerald ash borers flying around on this sunny Sunday morning. So we called it in. Oh, and nearby, ash trees planted along the roadside, turns out they were dead. The orange flagging was already there, and I don't know if somebody else had marked those saying, "These are dead, got to remove them." But they were infested, and they are now gone. A little bit more driving around town led us to find this tree on Washington Street, which is now gone, and the stump has been dug out since this picture was taken in December. And this looked to us infested even from ground level. It wasn't heavily infested, but it was removed. So soon after that discovery and the confirmation by Rick Hobeck at Cornell University of the identity of the adult beetles we collected, New York Department of Environmental Conservation had a number of infested trees destroyed. Since then, Melissa Firkey at Syracuse, State University of New York at Syracuse, has dated the infestation of some trees found this year at least 5 years. And last year, a whole bunch of the purple sticky traps were put out, but no beetles caught. But there is more trapping going on this year, and more infested trees were found in 2010. And along with Melissa, and actually it might be the next slide, so yes. Along with Melissa and Mark Whitmore at Cornell, and great cooperation from Jerry Carlson of the New York Department of Environmental Conservation, we're undertaking to put in a network of girdled trees as sentinels. And so I'll just say that a girdled ash tree, and maybe the next picture shows this, is one from which we've removed all the bark completely around it. They usually aren't this tall. This was a research tree. It really only needs to be a foot or so. That dooms the tree, but it will be intensely attractive to beetles, and they will lay their eggs in it, and the larvae will go to town. And so our plan with all of these girdled trees, of course, is to destroy them, cut them down during the winter or before next beetle emergence time, and peel the bark and quantify the attack. And hopefully we'll find a whole lot of trees with no beetles having attacked them at all. But I suspect that we'll find at least some. The other part of that program, I wrote tree girdling, sinks, and sentinels. So the sentinel tree would be a lone girdled tree. Well, near what we call ground zero, where we found the beetles for the first time, we've installed what is now more than a dozen sinks, and that's a group of 5 or 6 or 8 girdled trees all near to each other to create. The idea is to create a very intensive point of attraction for beetles in the nearby area, so that they'll attack those trees and not fly away. So if a newly emerging adult comes out, finds a hugely stressed tree, we want it to go to that stressed girdle tree and not fly away. So it's a management tool. We have no illusions that these trees are going to trap all beetles or limit the population in any significant way. So back to biocontrol, and in the context of what are the sustainable long-term solutions. We've already learned, thanks to the rather horrible process that people in the state of Michigan and elsewhere have gone through to try to save ash trees, that silvicultural techniques, that is, cutting down all the living ash wherever, within a half a mile or even more of where you find beetles, in order to prevent them from finding new hosts, that it's been shown not to work. Beetles fly too far, or perhaps more relevant, they're very hard to detect at low population densities. So they're already further than we can conceivably cut and remove all ash. So that kind of silviculture won't work, just like we know that girdling trees to make them attractive beetles won't work really to manage or control populations. Another long-term sustainable solution would be if we could magically make all ash in this country resistant. And so far, none of the ash species in this country have shown any resistance. There is some effort to save seeds of existing trees, to identify ash trees in areas where the beetle is occurring, identify those ash trees that seem not to be suffering as badly, that maybe do not die in response to attack by emerald ash borers. Maybe there is some native resistance. I think that's a long shot, but it's certainly worth watching out for. The other approach would be a classical plant breeding approach. And what has been done, for example, for the American chestnut is to breed into it the genes of chestnut species resistant to chestnut blight. Those chestnut species being those others that occur in Asia or Europe. And eventually, with enough back crosses, try to regain the character of American chestnut as a tall, columnar tree that plays a prominent role in our forests. With ash, a similar approach might, in the long term, work. It may be the ultimate solution, that we use related resistant ash or genes from it from other parts of the world to interbreed with our ash. That's a very long-term prospect. And then biological control, the reason that we're all here today, is another, we hope, shorter term, but still long term prospect. Um, so, classical biological control is, in a nutshell, you have a species that's a pest that's not native to a region, and that may be why it's a pest. It's over here and it's got none of its enemies. It's facing no resistance on the part of its hosts. So you go to where that critter is native, and indeed, emerald ash borer is not a significant pest of ash where it is native in Asia. You find why not. In some cases, again, it's resistant ash, but it might also be other natural enemies there, predators, parasites, pathogens. Uh, you test their safety and compatibility, release them into the range where this beetle occurs, and evaluate the impact. I've greatly oversimplified it, but what I hope this provides a useful segue to what Leah Bauer is going to present to us next. And Leah, I will turn it over to you. And I think we're just waiting for Leah to turn on her microphone.
Oh, I thought I had. Okay, hi. Thank you, John, for the introduction of biocontrol, and I do welcome the opportunity to share the work that I've been doing for the last 8 years. As emerald ash borer fell in our lap in 2002 while we were working on the Asian longhorn beetle in China, no less. So since then, we pretty much jumped the Asian longhorn beetle ship and have been working on emerald ash borer. And it's been quite a long haul. Before I move on, I did want to show you a close-up of the emergence holes, the D-shaped emergence holes that we had talked about, John had showed, and why they are D-shaped. So this is typical of Michigan forests. Many of you know Michigan is a bit of a swamp, and there's extensive areas of green and black ash in the lowlands of Michigan. Many of the ditches are lined with ash, roads. There are many sandy areas that don't have quite as much ash, but there is an extensive canopy of ash throughout Michigan. And you can see the characteristic symptoms. This is not the urban trees that John was showing, but this is probably at least 3 years into an infestation. The beetles tend to attack the canopy first, so you cannot actually tell they're there. Initially, the trees look very healthy. These trees probably had beetles working the midsection. You find them in the bole of the tree. And then they gradually weaken the tree and really don't attack the lower trunk until the very end of the last year of the life of the tree. It's a 3 to 5 year process depending on the initial health of the tree. So we've been, of course, wondering what to do about this problem. Initially, the interest was in ash removal, as you probably know, and a lot of money was spent in Michigan to try to create an ash-free zone around the lower 6 counties where it was first detected in 2002. And Canada did the same thing, except they used more than a half a mile. They had a 10-kilometer swath across the Windsor Peninsula where they tried to contain it to the Windsor, the west side toward Windsor, but the beetles had probably extended.
Leah,
Yes.
Leah. This is Peter. Your sound is breaking up.
Okay.
Did you? You've clicked the padlock in the lower left-hand corner. Is that correct?
Yes, I did.
Next to the talk button.
Yes, I have.
Okay.
I've turned it off. If I turn it back on again, would it
Okay, because
help if I
it's
talk
when your sound
louder?
comes through. When your sound comes through, it comes through okay, um, but it seems to be breaking up. Or maybe
Okay.
it's me. Two people just told me that your sound is fine. So,
Oh,
okay, so keep going.
all
Okay,
right.
it sounds like you're okay. Maybe it's
All
something
right.
here, but so you're doing fine. Keep going. Sorry.
Oh, no, that's fine. I I can't know unless somebody tells me. So, all right. So basically, the Forest Service being concerned mainly at that time about the forested areas, extensive ash stands throughout this part of the country as well as New York and Pennsylvania, Ohio. I know it gets a little less abundant in some states. So we started working on biological control in my lab and also some microbial control, as you know, with the work John's doing, as well as some work with a BT material that we're still working on. And that would target adults. That's quite a ways down the road, so I'm not going to even talk about it. Tree resistance is still pretty um, off way in the distance, although the BT gene is being put in trees um, through tissue culture methods to try to conserve these species for the future, hopefully. Conventional insecticides, we've had some real breakthroughs in the last couple of years with Deb McCullough and Therese Poland working with a product that Syngenta had sitting on the shelf. They didn't know what to do with it, but it happens to be a great systemic. It's imamectin benzoate. And the common name is triage. For those of you, it definitely has to be injected by an private applicator, so it is kind of expensive, but it is very useful for saving individual trees, high-value trees. John was touching on the idea of slowing ash mortality. The acronym is SLAM. Many of you have probably heard about this. It's a Deb McCullough product being where she and Therese Poland have put together everything we've learned in Michigan about emerald ash borer biocontrol and the attraction that emerald ash borer adults have to these highly stressed trees. And that's where the girdling comes in. So you can make sinks, bring them in, and either inject them with insecticide. Right now, this year, we're testing a bioslam where we're trying to see if we can help build up parasitoids more quickly in areas where emerald ash borer infestations are still at very low density, which makes it a little harder to get the parasitoids established. Quarantine, as you know, has had a limited success, and part of that is the inability of people to either know about emerald ash borer or I don't know if I want to say compliance, but I think they just don't really understand how serious the insect is and the long-term impact it will have. Um, let's see, I just switched to the next slide, but I don't see it. Do you see that slide? Okay, let's see if we can move to the next slide. Peter, are you there? I
I don't
can't
see
get
the next
this
slide
next
either,
slide
Leah.
up.
Click again. And,
Okay, here it is. Okay, I
okay.
did it again. All right.
All right.
So this, I don't know if all of you've seen this. It's a very appropriate comment, and I will emphasize to those of you working in the various states that to get out the word that you don't want to bring the dead tree from the backyard to your favorite campground. And that is a huge problem. It probably has resulted in many of the outbreaks in distant states, not all of them, but some of them, and certainly spread it around Michigan because everybody in Michigan likes to camp. So John gave a brief introduction. Biocontrol has been used for centuries, if not longer, where people have taken specialist natural enemies. I'll try to work my way through this with a pointer. And I do emphasize specialized because in the past that was not um, people didn't understand the risk of biocontrol, but we do have much better understanding of that. And the nice thing about biocontrol is it's sustained, so that you can actually, um, well, hopefully, if the parasitoids become established and are effective, that they're self-sustaining. You don't really have to go back in and continue to inject trees or spray. And generally, it focuses on non-native species that have been established for at least 5 years, cause economic and ecological damage, and where eradication has been shown not to be possible. And I think we've demonstrated that in Michigan and other states. So this is basically an outline of what I'm going to go through with you. Um, Basically, it starts with understanding the biology of the invasive species, and none of us knew much about emerald ash borer biology, although after looking at it and all these years, we now know it's very similar to the bronze birch borer and the two-line chestnut borer. It's one of the larger Agralis species. We also surveyed for native natural enemies, hoping there might be something we could take advantage of that was already here. The foreign exploration kicked in 2003. We found biological control agents. We have gone on to study them in the lab and in China, prepared the environmental assessment, got permits for release, and again have been working with it to look at establishment, and then of course the mass rearing, and now we're working on detection of these guys in the field and efficacy and uh, as it relates to ash survival. Now, this is a little bit of an overview of John's, but this is the reason I want to show you this is there is a wide window here of ovipositioning in the adults. They do have a 3-week window, but at least in Michigan, adults can emerge as early as May. So I have some ranges here. We've even seen some of these adults ovipositing still in September. So not only does tree health affect the life cycle of emerald ash borer in terms of the 1 or 2 year life cycle, eggs that are laid late in the season, those little larvae will overwinter as first instars. There's 4 instars. This picture over here shows you the 4th instar. And of course, this is the pupa, as John showed, and this is the egg. These are the 2 sites I'm going to talk about, the egg and the larval parasitoids. So in 2003, Dr. Ho-Ping Liu and I had been working on Asian longhorn beetle and started looking at the emerald ash borer here in Michigan and China. And these are the survey sites that we set up throughout southeast Michigan to look for natural enemies, both pathogens and parasitoids and predators. And what we found was less than 1% of the larvae were parasitized. And we also collected over 6,000 eggs and found one possible parasitoid, but it doesn't complete its life cycle. It pretty much dies as a pupa inside the egg. So there appears to be no egg parasitoids that are native or extant in the U.S. that are capable of attacking emerald ash borer. But these are some of the larvae. This one's exotic, Balca indica. These are actually we're finding now, at least in this part of Michigan, as the outbreak reaches the last year or so, Phasgonophora sulcata, this large solitary chalcid, it's the only endoparasitoid that's native. In other words, it develops inside the larva, its host. Many of these wood-boring natives, anyway, are more niche-specific than host-specific, apparently. And I think what they do is they build up on other wood-boring insects in the area. And we do find them this time of year walking the trunks looking for hosts. But at the time we did our work from 2002 to 2004, it was very rare in southeast Michigan. And it might be because we were more urban sites than we have up here, where we're actually able to observe them. This is a native Spathius. There's other native Spathius. We found other genera of braconids now, species of braconids. But again, they're extremely rare in most areas. A study was just published in Pennsylvania as well as in Ohio. And I would say that they're still just not able to key in to wood-boring insects such as emerald ash borer in ash. I'm not sure, they just don't have that coevolution. So the work in China, we were able to find all these these province or city provinces um, have that are highlighted here are the areas where we were able to find emerald ash borer anywhere in this northern part of China, except Shandong. Shandong province down in the southern part of China has a history of um, emerald ash borer, but for whatever reason, we weren't able to find it down there. It's probably they didn't want to take us to those ash stands. So ash stands throughout China have emerald ash borer, and they also have parasitoids. And the most widespread parasitoid we find in China, and these were new species that we discovered in '03 this little larval parasitoid. It's an endo, and it's gregarious, meaning that it develops inside, many individuals develop inside one emerald ash borer larva. And as with most hymenopterans, they're female biased, their sex ratio, um, and we get an average of about 70 adults per emerald ash borer larva. And they take about 4 weeks to develop in the lab, but in the field they do take a little longer. So here's a female right up here drilling in. There's a little pile of sawdust. Um, she's located it through sound and scent probably, and she's drilling in and she lays eggs inside the larva. And this larva actually sitting on some artificial diet here. It's not wood. And there's little swimmers in there. You can't see them very well, but these are the tiny little larvae, and as they grow, they consume the insides of the host, and they eventually break out and pupate right in the gallery, and they emerge from the tree by chewing a little hole, and they just swarm out. Oobius agrilli is the second parasitoid we discovered in China in 2004, and this one has only one little wasp that's about a millimeter long, and this is a newly laid egg, and one will develop in each egg. And these are parthenogenic, and so they don't actually need males. There are males, as you can see here. We occasionally find them. We just found one last week in the lab. It was quite exciting. But they don't need males, so we don't use them. We found it in the lab because we're still rearing out some eggs from China collection, but we don't use them at all for rearing. And we get a new generation every 4 weeks in the lab there with those little guys. And here's an oobius. Here's her ovipositor here. She's laying her egg right in the egg of the ash borer. And the healthy eggs look like this over here. Various stages of development. They turn kind of an amber color when they're mature. And when they're parasitized, most of them turn black. She can parasitize eggs until they're about 2 weeks old. They take about 3 weeks to hatch. If you were to crack one of those eggs open, you'd see a little chubby little larva in there. And they gradually pupate, and then the wasp will chew a little round hole out of the top of the egg and fly away to parasitize other eggs. Spathius agrilli is the third species. It's an ectoparasite of larvae. A little bit bigger. It's almost ant-like. It looks a lot like our native Spathius. So it does form a cocoon, a whole cluster of cocoons, and I'll show you that in a minute. But you get an average of 8 adults from each emerald ash borer larva. And they do develop more quickly, 3 weeks, in the lab, that is. So here's a female drilling in. Here's her ovipositor right there. And there's some eggs. The larvae live on the outside of their host, kind of like ticks. They attach on. And when they're mature, they spin a cocoon and pupate on the inside, and they also chew their way out of the tree. So during the last 8 years after we came to be able to lab rear these things, so the first thing you have to do is develop lab rearing methods for these insects. And the major concern was host range testing and locating other species to test. And it's not that hard finding other insects, but getting eggs of other agrellus species and other wood-boring insects was a challenge, but we managed to do that. IOBC, the International Organization for Biological Control, has a protocol, and they recommend you test 10 to 20 species that are phylogenetically related to the target pest. So you want to focus on those species that are genetically similar. So most of the species we tested were agrylus species. And then we went on to test buprestids and then other wood borers, insects that are in the same niche. And then we resolved some concerns that of threatened and endangered species and species of economic importance that Fish and Wildlife was concerned about. So I won't get into the details. This was our protocol for the two species that we have been working with exclusively in my lab. But basically, it's a stepwise process where you start with no choice tests just to look at physiological suitability and acceptance by the parasitoid. Then if you do get a positive, then you go to the choice test. And then if you get a positive again, you have to look at the risk-benefit analysis. You have to really make a decision. Is it worth? Is the known risk of releasing worth the benefit? So I did summarize or the potential benefit, because we don't know what the benefit is, but we certainly know the risk of doing nothing. So with Oebius, we tested 12 species, 6 agrylus, a couple cerambicids, and 4 different leps. We did have some acceptance only in the larger agrylus species, the 2 lines, the bronze birch borer, and the redneck cane borer. Most of our agrylus species are much smaller, and they don't even bother with those eggs. They don't probe them. They don't have any effect on the majority of our agrylus. So we did go on to choice tests, you can see here, and we found that they preferred overwhelmingly, almost exclusively, sticks that had ash sticks that had emerald ash borer eggs on them. They were all tested on their own host species. Tetrasticus, we tested 17 species. We had no crossover to other species. I think since then some more work has been done, and there is a little crossover, but is a very picky species, and of all these 3, it's the most difficult to rear. And being an endoparasitoid, we would expect it to be the most host-specific. Spathius, 19 species were tested. They did find 4 species were potentially attacked by the Spathius. They did not do the choice tests as we did. They did do olfactometry, and they found that Spathius prefers ash. And they also surveyed larvae in China and found that actually they collected agrylus larvae from many different host trees in China and found that this species of Spathius right here, agrilli, was not present in any other agrylus species in China, and neither was Tetrasticus. They did find other species of Tetrasticus and Spathius in China that specialize in other species. So the other study that Hopeng and I did in China was sampling seasonally. We had 1 year of funding basically for this, did 1 sample in 04, and then were able to do monthly in 05. And what we found out by calculating estimating the population reduction was that these 2 species on the green ash that was planted in this park in China, in this national park where we were working, it was a reforestation effort, that we can estimate about 74% of the population may be reduced by these 2, at least in the year we looked. And you can see that Oebius reaches about 60% of the parasitism eggs, and the Tetrasticus parasitizes about 40. But in some stands, if you sample through other sites, sometimes you get as high as 80 or 90% parasitism. So it just varies on site by site. Anyway, so in 07, we went ahead and applied for the permit, and that required writing an environmental assessment, which I have a link for it here. And you guys can go and read it, and it's long, but don't let it intimidate you. A lot of it is the maps at the very end that have the distribution of the 16 species of ash that are native to North America. Most of them are southwestern. In this area, we have the black, green, white, blue, and pumpkin. So we have 5 species. As you go south, you run into the Caroliniana ash. So potentially there's 6 in the eastern U.S. Those other 10 are all through the west. Most of them are in the southwest, and very limited distributions. So what we found was after 60 days of public comment, 30 favored releases, 11 opposed, and eventually APHIS prepared what is called a FONSI, a finding of no significant impact. And after extensive work with Michigan DNR and Department of Ag and faculty throughout the country, we had conference calls, and it was Michigan actually approved for release in the state. And so by the end of July, we were able to start releasing. And these were the sites we had. These little green triangles down here are where I was working with Oobius and Tetrasticus. APHIS, Julie Gould, who's the researcher out, or the entomologist out at the Otis lab in Massachusetts, has been rearing and studying Spatheus, and she has these 3 sites. And by the time winter was over, we'd been out cutting down our release trees and looking for establishment. And we found Spatheus had established at this site, and Oobius was found over here. That was in '08. And by 2008, we'd started to set up monitoring sites where we have at least a half a mile between a treatment area where we have release trees here in purple, and everything's being done on GPS. All these trees are being monitored for the long haul, and then a control site where parasitoids weren't released. By 2010, we've been able to expand in mainly because we've had huge demand for biocontrol from states where eradication has failed, or they didn't even bother. Like in Chicago, they just went right for biocontrol. I think by that time, the Maryland experience, where they'd done extensive efforts to eradicate, they were ready for biocontrol in '09, and we already have establishment of the 3 species down there, at least through one winter. And I don't know how they're going to do in the long haul at these sites. Those are a little more isolated, some of them. But up in Michigan, as you can see, these orange circles indicate areas where we've been able to document establishment of the 3 parasitoids, also this site in Ohio. Other sites, more recent sites, are pending. There is a lag time. And then more recently, it was very exciting here in Michigan. We were able to detect the movement of Tetrasticus way beyond our release site, 800 meters in 1 year. So it even went beyond the area of our control site where we're monitoring trees. So I think they're on the move. So right now, there is a biological control rearing facility that APHIS built in Brighton, Michigan, and that was completed in 2009. And they're already rearing the 3 species. And whoops, I didn't finish this thought here. Okay, so I'm going to talk about the Emerald Ash for Biocontrol guidelines that Julie and I and Jian Duan, who's an ARS researcher who's also involved now in helping us set up field experiments to evaluate parasitism and recovery and impact. But we put our brains together this winter and wrote these guidelines, and those are on the web, and I have a little bit of information on them here in this talk, on that particular guideline booklet for you here. But basically, all the sites are being monitored using handheld computers where you will enter the GPS data to track the releases and the tree health over time. And which species, how many, site characteristics, and then we'll continue to add establishment as we get to it and the efficacy down the road. This is the cover of, this is just a picture of the guideline booklet. I'm kind of wrapping it up here. I have a couple links down here to locate the guidelines. A lot of people have had trouble finding them. I know it's very difficult to read this page here, or table of contents, but I want to emphasize, don't let it intimidate you. This is all introductory stuff down to here. Just jump right down to there, and you can see the types of the plan that has been proposed for the states to, if they're going to be receiving parasitoids from Brighton. So, and just in a nutshell, a lot of people have wondered about the pre-release site preparation and how we're going to evaluate them. We've got it on a grid. We started out with a 200-meter grid with 50-meter grid cells. There's a plot epicenter, which is the intersection of, and it's larger grid. It's all on the computer, the handheld computer, so you can orient, and each cell will have an identifying letter/number in it, so you can orient it if it's a long, thin site, or in different directions. You can fit these 16 grid cells over the site. And then you actually pick 12 trees, ash trees that are infested in the center of the plot, and you release distribute the parasitoids on those 12 trees. And then these circles here that you see in each one, you'll just pick an area, a 10-meter circle, to identify the tree species and characterize the site in this 16 grid cell area. And here is a typical handheld. We use these little waterproof field resistant computers, and you just go into the field and we collect data on the species, the DBH. Of course, they're GPSed and tagged and flagged and crown condition, and I'll show you the 5 rating system that was developed at OSU with Anna Marie Smith and Dan Herms. Other information, this is probably for me, when I'm looking for EAB infestation, I pretty much look for woodpecker feeding. They're going to tell you where the EAB is, how far down the crown it's worked itself. It's working down by the amount of woodpecker feeding. By the time you see exit holes at the base of a tree, it doesn't have too many years left. Okay, this is Anna Marie's crown class. We just do a 1 to 5, 5 being dead, 1 being perfectly healthy. And I know you want to do 2.5 and 3.5 on some trees, but this pretty much sums up the life of the ash tree once you get into get the ash borer in your area. And we're hoping to slow that trend down. Let me just go back. The hope is that we probably can't change this trajectory once the ash borer comes into your stands. The hope is that the rate of increase of emerald ash borer, the next wave of ash borer, as the next generation of ash grow up from saplings and seedlings, that the parasitoids will be well established by then. I always tell people that my thinking about this, now everybody may not agree with me, but this is actually probably the best stage to get release in, get the parasitoids established for finding. This is stage 3. That we can, the hope is that these will become sort of parasitoid production units. And I know that sounds a little bit depressing, but you have to change your thinking on what is a tree. And an ash tree, we're going to have to look toward the seedlings, the saplings, the stump sprouts. And if you cut down some of those the pole-sized trees, and even a little larger, you're going to find that many of them do stump sprout, and maybe we can do some coppice management out there. But we do need to get these parasitoids established. So that's why we're testing some of these girdled tree zones for that. So this was a picture that a colleague sent from University of Michigan. The Michigan Botanical Club had been looking at this tree for many years. It's one of the state champions of green ash in Michigan, and you can see by 2009 it was gone. And so these are the kinds of trees we might want to, states who have large ash trees, they might want to consider injecting them. And I think that's it for my portion, John. I hope I left you enough time to wrap up your portion of the talk.
Thank you. Yeah, that's great. Let's see if we can, I'm going to minimize yours, minimize my introduction, and load up the brief thing I have on pathogens. Here it is. Drag it across. Oh, thank you, Peter. stepping in there. Wait for him to finish resizing. There we go. That'll work. So I'll finish with a short description of a research project that Leah and I and others that you can see listed there undertook in Michigan. It's still underway, but mostly done. And that is to test an EPA-registered microbial insecticide. The active ingredient are spores of the fungus Beauveria bassiana. It's labeled against ash trees and wood-boring beetles and all of that. And this fungus has been shown in other research in other systems to have the potential for synergism, where you could use a lower dose of one or the other of these agents with the chemical insecticide imidacloprid. That's sold and marketed in a number of under a number of trade names. And so, thanks to Leah for this gorgeous picture of an adult beetle that must have become infected while still alive as a newly formed adult, chewing its way out, but then died, and you can see the fungus blooming from within it. We worked with great cooperation with G Farms Nurseries, who had about 400 ash trees that in 2006 they could no longer legally sell them for shipment outside of the quarantine zone, which was much of lower Michigan at the time. And nobody in their right mind in lower Michigan wanted to buy an ash tree from them. And so they weren't in urgent need of the space these things were occupying either. And so they gave us 4 field seasons with them. And that was great for us. They're kind of in an orchard setting in 3 plots. Whoops, I went too far. I'm going back. So I mentioned the biological insecticide is sold under a trade name Botanigard. And we used the commercial product of imidacloprid, Bayer Advanced Tree and Shrub, applied as a drench once a year. The fungus we sprayed on. So there's the imidacloprid, that's Ho-Ping Lu. That's either me or Ho-Ping spraying the trunks. That's Mike Griggs spraying the crowns with the fungus. And we hung purple traps. We sampled beetles off of those traps to see if they were infected. That's the lower left there. We evaluated the canopy coverage and tree condition as Leah described in her talk. We counted exit holes. There we are. I vertically stretched this for its slimming effect on my physique there. And then we marked the exit holes from 1 year, so we didn't count them yet again the following year. We did a certain amount of work in the field, and then in our field lab equals hotel room to quantify fungus spore deposition and drift and persistence. We sampled bark, as you can see on the lower right, but taking little squares of bark and recovering viable spores from bark and leaves, and trying to quantify that. Whoops, I'm not getting good response on this. We found that the strain of fungus, the commercial strain is labeled on some of these slides as GHA. We found that it did persist in soils near there. There is some native Beauveria, some other strains of native Beauveria there. There we go. And we found, though, after spray, within a week, on the right-hand side of this graph, you can see the key, freshly sampled, leaves sampled freshly after being sprayed, leaves sampled a week later and 2 weeks later. We see a huge drop-off in the number. The y-axis there is the labeling didn't come through clearly, but it quantifies the number of fungus spores that we can find on those leaves. And within a week, there's a huge drop-off I'll show you some beetle data here in a second. But on bark, that drop-off is not nearly so significant. And the persistence is better. I mean, it kind of makes sense. Ultraviolet light is an enemy of these spores, and 10 minutes of exposure to the sun will kill a spore, or can. And so on leaves, they're more subject to UV exposure and they're more subject to wash off from rainfall. So it's a tough surface. We did beetle assays. With Leah's help, the lower left shows logs of ash that are infested with beetles being incubated in cardboard tubes so that the newly emerged adults can be collected. And then we use them in dishes, as shown in the center, by putting them on leaves that we collected from our trees from the field. In the lower right, you can see a cage that we developed. Mike Griggs made these cages to wrap around the tree, and we could put some beetles in there. They're very shallow cages. The beetles are kind of forced to crawl around on the bark, so we could control the exposure of these beetles to bark or to leaves. And the numbers in yellow, I'll focus on this table. Again, we sampled leaves freshly sprayed a week later and 2 weeks later. And the percent mortality, I'm going to try again to get my arrow to work. The percent mortality, there we go. On the imidacloprid treated, fungus alone is BB. Fungus plus the chemical insecticide at a low rate is BB plus IMID. And even at for leaves sampled in 1 week, my problem is when I try to move this arrow, I move the slides too. So the number 91 in the middle column there shows that was our evidence early on of potential synergy, the value of a lower dose of the chemical insecticide and the fungus. Okay, now I have instruction from Peter. Just click, don't drag. Yes, these numbers, within 5 and 7 days after a 24-hour exposure of beetles, we were showing the effects of the combination of fungus and chemical insecticide. So we did a similar thing on bark. There's no reason to think that imidacloprid would kill beetles, adult beetles placed on bark, because the fungus becomes systemic everywhere in all the tissues of the tree, and they have to be consumed. So the adult beetles aren't eating the bark, they are eating the leaves. So when the beetles are on the bark, we saw no difference between our fungus plus chemical insecticide or fungus alone treatments. No surprise. And so in our tree nursery, the emerald ash borer infestation increased rapidly. We found good when beetles land on leaves that have been sprayed with the fungus, they readily become infected and they will die within a few days. But if the leaves are allowed to sit in the environment for a week, they're no longer lethal as far as the fungus goes. So this has led to some follow-up work because of some of Leah Bauer's work showed promise that by treating the trunks, treating the bark of infested trees, you can infect adults crawling out on their way out. You may even prevent them from emerging. And in the upper right of this is a similar kind of picture that we took doing some work in a city park in Lansing, and the lower right shows the kind of emergence trap cages that we constructed to collect the beetles. So that's an experiment still in progress. The challenge we have near Lansing, Michigan is there aren't very many large ash trees left alive that are producing beetles, still emerging from these. Most of them are dead, and we've had to go farther afield to find trees to work with. And the other thing that we've started this last month was testing the compatibility of the fungus with parasitoids. And obviously the issue there is if we want to spray the trunks of infested trees with a fungus or the foliage, we need to be sure we're not also going to be spraying parasitoids and killing them. And so our first, we did an experiment 3 successive times this summer, and we're very pleased that the wasps seem not nearly as susceptible or not at all susceptible to the fungus at the kind of doses we were using that kill all of the beetles. So there is promise for some kind of combination. Okay, so I'm going to end with this little anecdote. This is a tough beetle, and it's tough to get around, and it is most likely spreading in firewood, as Leah pointed out. It's a huge public relations and public education issue. I think it's relatively simpler to regulate the nursery industry and the commercial forestry and logging industry, commercial wood. Uh, that's a lot easier, but regulating firewood and how members of the general public get around is tough. Uh, After one of our days of working in the commercial nursery in Michigan, we were driving from there about 30 miles back to Lansing, and we saw this emerald ash borer on the windshield of our truck. And I'll have you know that we are very careful and conscientious about washing our truck when we leave Michigan, taking it through the commercial washer, getting it hosed underneath, and getting everything off. But this beetle was on the outside of our windshield, and the speed limit is 70, so I was going 80. Oh, did I say that? And driving north, and this beetle hung on for a good 15 miles at 80 miles an hour until all of a sudden it just went boop and it was gone. And it's just my way of pointing out that the beetle is probably farther than anywhere we think, and we have to develop the tools to find it when it's in those places, so that we can see ash trees in our blue skies in the future. So I'm done, and I think Leah and I would be happy to answer questions for as much time as we have, Peter.
Okay, thank you, John, and thank you, Leah. That's a good presentation. Something was going on with my headset, so I wasn't having good audio, but it sounds like most other people were, so that's terrific. And I've recorded this webinar, so I will go back and listen to it. At this point, I'm going to close this window out, and I'm going to bring up, what is it I want to bring up? I want to bring up my exit poll. Here, and I'm going to make this chat pod bigger so people have questions. They can type questions into the chat pod, and we have a question from Jessica, looks like about woodpeckers, and I see that both Leah and John were also responding to questions as they were coming in, so that's great. John and Leah, as you see these questions, maybe you can just give an audio response,
Right.
and while people are doing that and typing in questions, please click the hot link at the center of the screen. That's an exit survey. It allows us to track the impacts of this webinar. And there's also a place where you can type in questions if you have an additional question that you think about later, and we'll try and get responses posted to those questions. So feel free to start. Let's go with the questions, and either John or Leah or both can respond. There's one there from Jessica about the woodpeckers.
Okay, that's fine. It's not a silly question, of course. Woodpeckers are happy to eat emerald ash borers, but they couldn't possibly keep up. And they can't respond as a population as quickly. If the forests were continuously infested with high populations of emerald ash borers, we would certainly eventually have higher populations of woodpeckers. But they can't. The insects will quickly outnumber them. So they are a good indicator. They're not really a biological control agent. And then the next question from Talbot Trotter states simply the impact of fungi on natives. So this particular strain, with microorganisms, the strain matters, not just the species. With this particular strain, it's commercially registered. It has a relatively broad host range, but it has been tested and registered with EPA. So therefore, it had been tested with a certain set of non-target studies that are required. For example, it would have had to been shown in typical use patterns for the insecticide, like spraying in an orchard or a nursery, to be uh, compatible with important non-target things like honeybees, for example. And so that it is important for us to test it against the parasitoids, and that's why we're undertaking to do that. I hope that gets the question you're answering.
Okay, so I have
And so,
a
yeah,
question here on at what point does biological control become a success? I don't think we know enough about the variety of infestation levels and also the density of ash. The forest is so complex. You've got stands that have very few ash trees. Emerald ash borer does tend to find those. I'm not sure if we're going to be able to get the parasitoids established in those areas as readily. So I I do see a lot of ash coming back in southeast Michigan already. And so and the ash borer is still there. So we don't know if we can get the parasitoids established in this aftermath forest, what I call the aftermath forest. We're hoping to catch the population of EAB on the upswing to get the parasitoids in such that we can use those standing trees to produce these parasitoids. And hopefully they'll suppress, if you think about waves, so you've got the interval and the height of each peak, hopefully we can bring down the density and bring those peak population densities to lower levels and spread out the intervals of these outbreaks so that the ash trees can tolerate them, because our native ash trees, although they are more susceptible than the Asian ash species to ash borer, and much more attractive certainly, they can resist, and they also can survive a certain density of attack. So that's the good news. We just have to bring that density of ash borer down to a more tolerable, to a threshold density that ash can survive. And different species, of course, have different um, susceptibilities. So the first ash species, unfortunately, to die are the black ash. They're the most susceptible. And then the green is the next. Then we've got the white, and then the blue is the most resistant. So it's complex, but I think we're going to see a lot of changes in our forest over the next couple generations of people. Okay, was there another biocontrol question in there that I saw? Oh, how many acres of land for native wasps? That's TACE ref, and I don't know whether nobody's working on at least, there are some people working on a native wasp. But again, it's kind of like woodpeckers. We're not sure if it's going to be effective. So far, it hasn't been effective. It comes in kind of late in the infestation cycle, and maybe moving over from species of Agralis that it naturally attacks. Oh yes, I can send you the cartoon. I don't know that it's a poster, but I can send you that cartoon. I'm not sure where it came from. It's been floating around cyberspace with the camper. It's a good one. All right, what types of stands would be ideal for introduction of parasitoids for long-term effect? Well, I do think that stands that have, I, at this point, I look for stands that have ash borer in them. But it's likely that most of the stands that we think don't have ash borer in them because we can't see the overt symptoms, it's already there. Usually by the time these sites are discovered, later on people track back or they do the dendrochronology, and they find that the ash borer has been there 5 to 6 years already. It's a very elusive insect to detect, and I do think that it's going to be, if you're in a vicinity, and John can attest to this because of the Randolph situation, the further out they go, the more they find. And that was true of Southeast Michigan too. We
yes,
started out with 6 counties, And they kept adding these rings of counties until they realized that the whole lower peninsula was infested because of campers taking firewood off to the campgrounds.
Yeah, the beetle on its own, the individual beetles have been shown in flight mill tests with a colleague of Leah's to fly as much as maybe 20 miles. I think that's probably rare in nature, but the fact is some of them are capable of it. But on its own, a couple miles a year for a given population to kind of spread outward is reasonable. But it's humans. It's humans moving firewood in the trunks of their cars, the principal means at this point that these beetles are getting around, and it's leapfrogging any kind of population management efforts we might undertake. So let me get at a couple of these other questions. Is ash borer a problem on the same scale as the Asian longhorn beetle? And there, the issue is just the very different biology of these critters. Asian longhorn beetles aren't as active or long distance a flyer. I think some emerald ash borers are born to fly and will zip away. Asian longhorn beetles take longer to kill an individual tree. On the other hand, Asian longhorn beetles have a wider host range. Sugar maple is they love it very much, but they'll also do just as well on other trees. Emerald ash borer only ash, green and white and black being the principal ones in our forests. Is it on the same scale? It's on a bigger scale already. It's already killed many times more trees than Asian longhorn beetle has. That being said, I said sugar maple. Think how important sugar maple is to our forests. It is a keystone species in northeastern North America, anyway, and more important relatively to the forest as a whole as ash. That's a tough statement to make, but there's a question about if biocontrol is a viable approach in urban areas. Go ahead.
Oh, I wanted
Leah,
to
were you
just
gonna say
mention,
something?
and John, I don't know if you mentioned this, that Asian longhorn beetle has a broader host range. So it cuts across genera and
Right,
families of trees. So it's not just sugar
right.
maple, it's pretty much every hardwood that's out there, except possibly some oaks. It wasn't clear that they can complete development some oak trees. I've seen them in China and they grow mostly North American species in the areas of China I've been. They've lost a lot of their seed source there, and these species have built up, and they turn them into crates and pallets. And so we're going to find
Right.
more pockets of Asian longhorn beetle, but the good news is that it's a large enough beetle. If it's on people's radar screen, they're going to hopefully they'll notice it more quickly than they did in Worcester, Mass, where it had been there for years. But the Jamaica Plains location, as well as Chicago, they've been able to. Well, Jamaica Plains is so new. I don't know how that eradication is going to go. But Chicago, they may have successfully eradicated it. But again, there's not as much forest out in Chicago. It's very chopped up with fields in urban areas.
So, Leah, one question there is the question from Virginia Mayo Black. Any indication of biocontrol is a viable approach in urban areas?
Oh,
And you
for
can
emerald ash
follow on to
borer.
my first.
Right. Okay. So
Yes.
I could obsess over Asian longhorn beetle too, but I I don't think so. It depends on what kind of quality of tree you want. The green ash that we see in China, it's under a lot of pressure from EAB, but the condition is it's not a lovely tree. But if you had some woods in the back, you don't mind some trees with cankers and maybe a dead limb here or there. You'd have to do more maintenance on them because of the hazard. And in other words, removing dead limbs. It's not clear to me that it's going to have a huge impact until the first wave of it goes through and most of the big trees die, because the big trees in the forest are pumping. The more phloem you have, the more beetles you produce. So you're just pumping a huge number of beetles. We'll never have this big front again. So as these big trees die off, A lot of trees are going to die. So I think through this first wave, I would recommend if a tree is large, and Cliff Sadoff at Purdue has a really nice software program on the cost-benefit analysis of it's called a ash cost calculator. Should you treat or not? And it's very interesting because the bottom line is a big tree takes a really long time. You'll never see a big tree again in your lifetime. It costs more to treat a big tree, but if if you have the money, it's worth saving a big tree. A small tree that was planted maybe in the last 20 years or even 30 years, probably worth replacing. Unless you just really love that
So
tree.
one of the so the issues in urban areas are if there are large trees for shade, the biocontrol agents are not going to keep emerald ash borers from attacking that tree. They may be doing the biocontrol agents may be doing just fine and reproducing and have their own viable population, but they're not going to kill all the emerald ash borers, and so some of them will be alive to kill at least parts of those trees. And there are, as Leah points out, viability issues. So it would definitely be possible to get biocontrol agents established in urban areas, but it may not be an approach to preserving the ash trees in those areas.
Yeah, they're
I'm going to
it's
get to
the
some of the other
hazard is
questions.
huge. The hazard of
Right.
these trees in parks and along streets, unless you're going to be injecting them with
So,
Benben.
there's a question: is the recommended application of either spray, drench, or injection? There are a number of pesticides been tried in all of those ways for emerald ash borer control. The drench is what we use because that's the kind of thing a homeowner would do to preserve their tree. And on a relatively small tree, not a huge tree with complicated architecture, relatively small tree, I believe that drench of imidacloprid could work to protect that tree for quite a long time. The problem is you have to apply it every year, and it adds up. These are probably trees in people's yards that they've never spent any money on, and now you have to figure out, is it worth spending $100 a year on a tree? Injection is the most reliable way to get an insecticide into the tree at a useful dose, and the insecticide that the folks in Michigan have done a whole lot with, emamectin benzoate is the chemical name, is applied by injection. And that's a huge disadvantage because that's a costly undertaking. But the advantage of that insecticide, at least so far, is it looks like a single injection um, can last for at least 2 seasons, possibly 3. So you don't have to apply it every year. But it is expensive. It may be a way to preserve the higher value trees. And so that's another webinar, is chemical control of these beetles, and it's a little outside of mine or Leah's expertise. There's a question from Brian Keevan about the 80-mile-an-hour story. Is it already here in Massachusetts? It very well could be in Massachusetts. But probably not, because it hung onto somebody's car at 80 miles an hour on the outside. It probably because it traveled in somebody's car in firewood in their trunk or in the back of the truck. If it's there, that's how it got there. That's my professional prediction, I guess. But let's hope it's not. And the other unknown that we have in western Pennsylvania and western New York is that white ash in the forests, and green ash too, are a relatively large component. It's still only 7 or 8% overall in New York, for example. But I'm sorry, I should have said the forests are a relatively large component of the landscape. The contiguous forest acreage in western Pennsylvania and western New York is huge. So the beetle on its own might get slowed down because it's finding so many hosts in this friendly environment. But it's the human movement that's going to that we as humans are at risk that our conspecifics are going to be moving this out in the trunks of their cars to western Massachusetts or to the Adirondacks or further north into New England.
Kim, I have an opinion on gypsy moth versus EAB. Um, I think gypsy moth has been certainly a destructive force in terms of weakening a large number of hardwood and conifer trees um, throughout the eastern forests and occasionally in the west. Um, but certainly, it doesn't have the immediate effect of killing trees. Gypsy moth seems to be more of a people problem. A lot of people don't appreciate 100% defoliation of their forests, just as the leaves flush, and that does weaken the trees. But um, again, it's in the black caterpillars coating the hairy caterpillars coating the sides of their houses. I mean, and we do have nice fungal control of gypsy moth now with a fungal pathogen that is from Asia, and it's an incredible biocontrol agent. Gypsy moth, in general, is a non-pest in Michigan in the last few years. So, but there are control approaches now. They do have BT in the periodic outbreaks in dry years when the fungus doesn't impact gypsy moth. So, I think if we can continue to do more research on emerald ash borer, we can turn this around with some of these natural enemies, either pathogens or things like BT. But it's going to take some time, and certainly slowing the spread of gypsy moth has been very beneficial to states.
There was a question about hemlock woolly adelgids, and again, I'm not an expert in that, and I do see that Mark Whitmore has signed on to this webinar, and he's at Cornell University, and I know he knows and works on hemlock woolly adelgid and its biocontrol. So that's the same department Peter Smallage is in, the Department of Natural Resources at Cornell. I'd suggest contacting him, and I'll bet he'll be happy to answer that question about the hemlock woolly adelgid. Okay, I for one have enjoyed this tremendously, but I got to move on to lesser and less interesting things, and have to sign off and say goodbye to you all. And I'm sure Peter can wrap us up here, right?
Okay. Yes. Thank you, John, and thank you, Leah, and thanks to the audience. We had almost 60 people hang in here for an extra 30 minutes, which is remarkable and a testament to the quality of the presentations and the interest that everyone has in the subject. The webinar was recorded. I'll send a note to the webinar list to indicate the location of that recording so people can go back and watch it. And Leah and John will join me again tonight at 7 p.m. for a live presentation. I'm going to try a different headset so I can hopefully have better audio. But my thanks again to Leah and John and to the participants, and I hope everyone has a nice afternoon. So
bye all. Thanks for tuning in.
Thank you all.
Bye-bye.